Now we will try to introduce Professor Olsen, who was born in Norway in April 1940. He received his medical and doctorate degree from the University of Oslo in 1967, where he becomes a faculty member of the Anatomical Institute and conducted molecular study on the structure of collagen in 1971. He moved to the United States to work with the Professor Darwin, and 1 year later joined the faculty of the Department of Biochemistry at Regressed Medical Schools. In 1985, Professor Olsen was appointed the Hersey Professor of Anatomy and Cellular Biology at Harvard Medical School. Now Hersey is at cell biology in the same department. Since 1996, he has also been senior member of the staff of Forestry Institute and professor of the Department of Biology at Harvard School of Dental Medicine, where he was Dean for the search from 2005 and 2017. The floor for you, Professor Olsen.
I feel honored for staying here, and I hope you will like what I will tell you about the skeleton and how it works. Now, obviously, the skeleton is absolutely essential for us to sit here, and it's remarkable how the cells that form the skeleton are able to put this wonderful organ system together during development and change it as we grow, and then finally keep it going until we are very old. And as a medical student, I was many years ago really fascinated by the molecular biology, the cell biology, the genetics that really makes this possible. And I decided that as a scientist, I would take advantage of the mutations that occur in different proteins and different genes that affect the skeletal development and function to really understand the mechanisms, the molecular, the cellular, and the biochemical mechanisms that makes this possible. And many years ago, in 1996, I had a postdoc in the lab, Matt Warman, who learned about a disorder called Osteoporosis Pseudo Glioma syndrome. And these are individuals that have too little bone, almost no bone in certain places. And this is combined with blindness. And by getting patients with this syndrome from many different collaborators, we were able to map the position of the mutant gene in a specific chromosome, chromosome 11 in humans. And once we had mapped the position of this gene that made formation of the skeleton impossible at certain places, and you can see here below, on the left side, at the very bottom, you see the bone, the pelvic bone in a patient with this disorder is very thin, like paper, compared with above it, the pelvic bone in a normal individual. And we were eventually successful in finding the gene that causes this syndrome of loss of bone or very little bone. And this gene coded for a receptor that sits on the surface of bone-forming cells. It's called LRP5, and you can see that. And this is a receptor that binds secreted glycoproteins called Wnt proteins and a frizzled protein that sits on the cell membrane And when LRP5 forms a complex with this Wnt protein and the Frisell protein on the cell surface of bone-forming cells, then the cells become osteoblasts, or bone-forming cells. So this decided that we wanted to understand what the downstream signaling was and what this would do. And so this is a case of osteoporosis, genetic, that is caused by this LRP5. Now, it turned out that LRP5 also could be integrated and form a complex with another protein called sclerostin. And this sclerostin, when it binds to LRP5, prevents LRP5 from forming the complex on the right and signaling an osteoblast formation. So in the cells that produce bone, there are LRP5 signaling pathways that form bone, or just, or as you can see at the bottom. But in addition, cells producing sclerostin put a brake on the activity of LRP5 and bone-forming cells. And this sclerostin makes certain that we do not have too much bone, but just the right amount of bone. And this was discovered at about the same time, that with mutations in sclerostin, so that sclerostin cannot bind to LRP5, then you end up with individuals with too much bone. So much bone, in fact, as you can see on the left, on the bottom, that one skull of a patient with mutations in sclerostin leads to sclerostosis, and a bone in your skull that is as heavy or heavier than 3 normal skulls. So here we have, with mutations in LRP5, you get too little bone. With mutations in sclerostin, you get too much bone. And this has led very recently to a new drug to prevent or use in osteoporosis. Because it was clear from this that if one had an antibody that would bind to sclerostin, normal sclerostin, and prevent its binding to LRP5, one would have a drug that would increase bone formation. And you may know, if you're interested in bone biology and osteoporosis, that this drug, an antibody to sclerostin, is now approved for use in patients in the USA and in Japan. And it replaces many other drugs that have been used for a long time for treatment of osteoporosis. Now, at the time when we discovered some of the pathways that lead to bone formation, we also discovered in patients that bone can go haywire. Bone formation can, together with inflammation, really destroy bones. And this mutation I show here leads to a disorder called cherubism. It's a genetic disorder with excessive inflammatory bone loss in children in the jaws, in the mandible and the maxilla, and in not other bones. And it was clear to me that if we could find the mutation that resulted in this disorder, we would learn something specific about what controls the formation of the jaws that we all have, the mandible and the maxilla. And in this disorder, you can have familial and sporadic cases. Most of the disorder, the cherubism, are produced by dominant mutations, but there are rare recessive cases. And the onset of this disorder starts pretty soon after birth, around 1 to 2 years of age. So in the period between 2 and 4 years of age, this disorder takes place. And remarkably, after puberty, some of the effects of these mutations regress, leaving some facial deformity or radiological evidence. So again, having these patients, finding the gene would tell us something about what controls the bone formation in these specific bones in the jaws, as well as what would happen during aging when these children with these mutations would grow up. And if one looks at pieces of tissue from patients with cherubism, you also see, as shown here on the right top, there's inflammation in the tissues that normally should have the bone. And within these inflammatory fibrous tissue regions, you see a little bit of bone, and you see multinucleated giant cells. And these giant cells are remarkable. You see them here in the hard modification. These are cells that have 5, 10, 20, sometimes 50 nuclei in a single cell. And these are not the normal cells we have in our bones. And as a consequence of these cells, the bones in our jaws are eaten up by these multinucleated giant cells, the osteoclasts, bone-eating cells. Now, it was known at the time when we looked for the mutations responsible for this disorder that myeloid cells in the bone marrow, shown here from the very bottom and up, form the cells that over time develop into macrophages and osteoclasts. And for the formation of macrophages and osteoclasts from myeloid cells in our bone marrow, there are certain cytokines and receptors that must be activated. And so a macrophage colony stimulating factor, MCSF, is needed to form the macrophages. And together with that cytokine, we need another molecule called the rank ligand to bind to a rank receptor on the cells to form osteoclasts. And we were wondering whether the mutations in cherubism could actually explain what the connections might be between these different steps in the formation of osteoclasts. And also tell us something about how bone marrow stromal cells regulate the formation of macrophages and osteoclasts. And it turns out that in addition to having factors that can produce osteoclasts and macrophages, stromal cells in the bone marrow produce proteins. Osteoprotegerin is one of them, that prevents that process. And again, this is a system where you have cytokines that stimulate osteoclast formation, bone resorbing cells, but also cytokines and molecules that inhibit, again, to make sure that we do not get too much osteoclasts or too little, just right, as if you have in the system that produces or stimulates the production of bone by bone-forming cells. So we were able to map the gene for cherubism on chromosome 4, at the very tip of chromosome 4. And it turned out that that part of chromosome 4, when we discovered this, had already been sequenced. And the sequence was quite good. But in the region of that gene, we had 25 genes. And so to find the gene that was mutated in cherubism, we started sequencing one gene after another. And that took some time, but we identified the gene in 2001. And we had 15 families with cherubism, mostly from Brazil, but we found mutations in 12 of these families. Now, these mutations were in the protein that had the not so interesting name, SH3 binding protein 2. It's a protein that is produced by a gene with 13 exons and separated by introns. And all the mutations that we had in these patients were mutations that changed an amino acid in exon 9. And the mutations in all these families we looked at changed an R, P, G to something else. R for arginine, or proline, or glycine, very close together within the sequence of 6 amino acid residues in this protein. And they were replaced by either glutamine or proline, leucine, arginine, histidine, etc. And we did not find any mutations elsewhere in this protein. And it turned out that when we looked at the protein, it had many different domains that could interact with other intracellular proteins. But these domains were not carrying mutations. All the mutations were in one region, outside any region that looked like the regions interacted with other proteins. And so the question then was, how could these mutations affect the functions of this protein? Well, the more we looked at the protein, we more found that it really is interacting with many different proteins in practically all cells, but especially in bone-forming and bone osteoclast-forming cells. And its position or its activity activates multiple signaling pathways in the protein, in the cells that express this SH3BP2. And we then decided that in order to understand how it works and what the mechanisms are really for cherubism, we had to make a mouse model. You cannot do experiments with humans, so we tried to do it in the mouse. Put one of these mutations that we found in the patients into mice. Now, in doing that, many people working with mice at the time would almost immediately knock the gene out. And I'm very glad that we did not do that. Because what we did find was that there's another syndrome, Wolff-Hirschhorn syndrome, that have deletions in chromosome 4. And frequently, the deletions in this syndrome include the gene SH3BP2. And none of these patients had cherubism. So clearly, we should not knock out the gene for SH3BP2 to get a mouse model for cherubism. We had to take one of the mutations in cherubism from humans and put that type of mutation into the gene of the mice. And we did that. And that gave us a mouse with cherubism. Except that when the mice carried only one allele with a mutation, they were losing bone, but they did not have inflammation. However, when they were carrying two alleles of the SH3BP2 with that specific mutation, their whole body turned into an inflammatory bone-losing structure, as if the whole body of the mouse was a copy of what happens in the jaws of people with only one allele mutated in this protein. And so, what happens in the jaws of people with cherubism happens in the whole mouse with carrying the mutation in the two chromosomes of SH3BP2. If we have one of the two alleles mutant in mice, we get aggressive giant osteoclasts with multiple nuclei, but we do not get inflammation. So we have looked at these mice. As one would expect, the mice carrying the mutations in both alleles on the top right lose bone. And the bone loss is caused by osteoclasts that are abnormally large. They have, instead of 4 or 5 nuclei per cell, they have up to 50 or even more nuclei in cells. And these cells sit on bones, and they really eat the bones up in the mice. And as you can see here at the bottom, in the mice, there are increased numbers and increased sizes of osteoclasts, going from the wild-type control on the left to the double mutant mice on the right. Now these osteoclasts are not only containing many nuclei, but they're giant cells. And that can be shown in looking at mutant bones. This is the maxilla of the mouse, cherubism animal. And on the right, you see in dark color here, these osteoclasts that are sitting on the surface, and they are absolutely abnormal. And not only are they abnormal in the mouse, but when we take bone marrow cells from normal or mutant mice and add the proteins that are needed for their differentiation to osteoclasts, then we see that in mice or in cells from mice with cherubism, we have these gigantic osteoclasts. And this you can see better here. So on the top, these are normal myeloid cells stimulated to become osteoclast. And you see the osteoclast on the top here. They're very small cells compared to the mutant cells. Mutant cells are gigantic, and the number of nuclei really is proportional to their activity as bone-eating cells. And not only are these cells eating bone, but the inflammation and macrophages in inflammation start eating bone from the outside, not only from the inside. So in cross-sections of a femur of the control mice and mutant mice on the right, when you have 2 copies of the mutant SH3BP2 in mice, the bones are eaten from the inside by osteoclast, but also from the outside by inflammatory macrophage cells. And this is in the mouse. It's not only macrophages that invade and eat bone, but there are macrophages that go through all organs, including liver, lung, and stomach. And what you see here on the left are in these different tissues staining for macrophages that have invaded the liver and the lung and the stomach in the mutant mice. And what we discovered was that not only are these tissues invaded by activated macrophages, but these macrophages produce tumor necrosis factor alpha, TNF alpha, in large amounts. Normally in mice, you cannot detect this in the serum, but in the mutant mice, shown here on the right, you have very high levels. So these macrophages that invade the organs and eat the bones spell out really large amounts of TNF alpha, and as a sign of highly activated macrophages. And these highly activated macrophages invade joints that destroy joints, shown here in the elbow joint, comparing the wild type and the mutant joint, and the knee joint, as if you would have absolutely no respect for the bones here, but cells that try to, as much as they can, destroy the bones and the joints between the bones. So this led us to put together a hypothesis for what mutations in SH3BP2 can do. It happens in myeloid progenitors, and because of the mutation, these progenitors become overly sensitive to incoming signals. And you get activated, highly effective, and TNF alpha producing macrophages on one part, and that leads to inflammation and bone loss. In addition, you get hyperactive osteoclasts coming from the same myeloid cells that really, on top of that, destroy the bone. So it's said that SH3BP2 is important for the balance and the response of incoming signals in myeloid cells and the formation of macrophages and osteoclasts. Now, we looked at in the mice, we could, of course, determine some of the signaling pathways that were affected by these mutations in osteoclasts and macrophages. And in doing that, we found that TNF alpha production is up because of specific signaling pathways, ERK1/2 pathway, for example, that stimulates TNF alpha production. And since we had this high TNF alpha production, we obviously thought that if we knock TNF alpha out, we remove that gene from the mice, perhaps they would be normal. And we did that. We took the TNF-alpha gene and produced mice that were deficient in this gene, TNF-alpha, and we improved the bones of the mice. However, what we did find was that the invasion of macrophages into liver, lung, and stomach, shown here, without TNF-alpha, disappears. The inflammation disappears. However, when we look at osteoclasts, there's no difference whether you have TNF-alpha gene active or not. So in the diagram that I showed you, that SF3BP2 in myeloid cells leads to macrophage activation and osteoclast activation. If one takes TNF-alpha out of the mouse, then the macrophage arm is normal, but the osteoclast arm is not. So there we looked at what determines the high activity of osteoclasts. We found that downstream of the mutant SF3BP2, there's a protein NFAT-C1, which controls osteoclast differentiation. And this was upregulated in the mutant mice. And NFAT is an inhibitor of the osteoprotegerin that in turn reduces osteoclast differentiation. So with NFAT-C1 being up, then osteoclast differentiation would go up. So then we decided that we would simply knock NFAT-C1 out and see whether that would take care of the osteoclasts. And in fact, that happened. In these mice without NFAT-C1, we had no longer osteoclasts seen on the right here that are abnormally large and very active. We have normal osteoclasts. However, the invasion of inflammatory cells, macrophages, into the lungs or other organs is now not normal. So we have two arms, and we can play with proteins within the two arms to reduce the bone loss in cherubism. But then, with all this, we still do not really know why mutations in a very small region of this protein can affect all its interactions with other proteins and signaling pathways. How could that be? And these mutations are in a very tiny portion of SF3BP2. And it shouldn't affect any other part of the protein. And all the other parts of the proteins that are seen in green and red here in the diagram, these are the parts that really stimulate signaling pathways in cells. So what is these mutations on top here? What are they doing? And this was solved with 2 papers in Cell, showing that this protein, like many proteins, they can bind to tankerase. Tankerase is a poly-ADP ribose polymerase, and it binds to specific proteins in cells. And when it does that, it puts a signal on other parts of the protein. And in this part, it's one of the parts that are involved in signaling pathways, interactions with SF3BP2. And this signal put in by binding of tankerase to SF3BP2, that leads to ubiquitination and destruction of the protein. So what is abnormal in these mutations in cherubism is that it is not degraded. It builds up in cells, so the level of expression of this specific protein is too high. So that what really causes cherubism with these mutations in SF3B2 is that there's too much of it. There's nothing really wrong with it, but it's simply too much. And so the region in SF3B2 that allows it to bind to tankerase, the question is that region shared with other proteins? And in fact, one of the two papers shown here shows that there might be 200 or more proteins that carry a small region similar to the region that has mutated in cherubism in SF3B2. And that in these proteins, any mutations in this kind of small region might again affect its binding to tankerase and therefore destruction, which is the normal way of making sure that the levels of the proteins are not too high and perhaps not too low. And so, having done this, we still were wondering, why in humans are these mutations primarily affecting the jaws, the mandible, and the maxilla? And so, looking at what happens from birth with these two bones, both the mandible and the maxilla at birth are simply rod-like shaped proteins. Within 4 to 5 years, these proteins are remodeled enormously. And to remodel the protein and reshape the protein, you have to degrade the protein, the osteoclasts, and activate osteoblasts and form bone. So at 4 to 5 years of age in humans, these two proteins are the only proteins in the skeleton that are almost fully sized and shaped. And so this activation of these specific proteins might be one of the reasons. But there might be something else. And that is that cherubism, when it occurs in patients with mutations, happens mostly before 5 years of age. Then it starts. And what is unique, in addition to the remodeling early in life of the jaw bones compared with other bones, is that these two bones have macrophages that are responding to oral bacteria and tissue fragments caused by jaw remodeling and tooth eruption. And in a paper with my postdocs and other collaborators as well, we published in 2014 this cell report paper showing that what initiates the cherubism happens in the jaw bones because the macrophages are activating with the mutation SF3B2. They activate what is called the toll-like receptor, the immune system that we all are born with. And what happens here is that in addition to having macrophages on the left here, TNF-alpha producing macrophages with a mutation in cherubism, these macrophages in the jaw bones increase the amount of what is called PAMPs or DAMPs. These are organism and jaw tissue fragments that can bind to the toll-like receptors shown here in the red. And by binding to these receptors, you activate another signaling pathway in macrophages that produce more TNF-alpha and add to the process. And within this part that really develops the cherubism phenotype, there is downstream of the toll-like receptors a protein called MyD88. And if this is correct, one should expect that by knocking out MyD88, that one would reduce the effects of cherubism. And this is exactly what happened. So now we made a mouse that had not the cherubism mutation only, but in addition lacked the MyD88 shown here. And in these mice, the TNF-alpha production goes down to 0, normalized. The effects of liver, lung, or macrophages getting in and causing inflammation and inflammatory loss of tissue, that's normalized. So clearly, for cherubism and the mutations to really work, certainly in mice, and we think in humans, you need, in addition to the original mutation in the macrophage, you need the toll-like receptor activities in the jaw of the patients. So that leads me to the last question I want to address, and that is, why do the disorders of cherubism regress after puberty in humans? Now, it turns out that this toll-like receptor effect in macrophages in the jaws, not only do they have MyD88, but another protein, IRAK4, with mutations in MyD88 or IRAK4, you get in humans another disorder called primary immunodeficiency. And this is particularly susceptible to pyogenic bacteria. Now, in people, not with cherubism, but with this primary immunodeficiency, after puberty, as they grow older, it doesn't seem to matter anymore. They don't die. If they die of the immunodeficiency, it happens before they are 11 or 14 age. And so afterwards, after puberty, this system involving toll-like receptors and downstream signaling pathways seem to be turned off. And I think that is a good chance that this is why in patients with cherubism, you get regression after puberty. This, of course, has to be studied, and we're not kind of do that in mice, but you have to do it somehow with patients. And finally, do these mechanisms point to effective therapies for patients with cherubism? Yes, I think we do. So in looking at the critical parts in the signaling pathways that cause the cherubism disorder, you can affect the TNF-alpha production, number one. You can affect rank ligand and the formation of osteoclasts, number two. Or you can affect NFAT-C1 that has to do with differentiation of osteoclasts. And all three things have been tested in humans. And in fact, it turns out that antagonists of TNF-alpha, rank ligand, and NFAT-C have been tested. And outcomes is that for number one, TNF-alpha is not sufficient. It doesn't really fully work in patients. However, if in mice you take away TNF-alpha at the very beginning of the mice, before they get cherubism, that prevents it. But that's hard to do in humans. The rank ligand effect have been tried. It doesn't really work either. It doesn't really cure cherubism. However, affecting the NFAT with a calcineurin inhibitor, calcineurin is absolutely essential for the effect of NFAT, that has been very good. And in fact, that you can see here, before treatment on the top left, next to after treatment of a 4-year-old boy, for 1 year on the therapy, it's almost rectified. So I think we do have now a way of treating patients with cherubism. Now, all these studies, starting from the very beginning, I would say is due to 3 postdocs, Yasuyoshi Ueki from Japan, Valdenise Tiziani from Brazil, and Ernst Weichenberger from Germany. Now, Ueki got his PhD degree in the laboratory of Professor Yoshifumi Ninomiya at Okayama University. And Ninomiya was my first postdoc when I came from Norway to the USA. And he returned then to Japan. So he is my or he is my son, Ueki. And in addition to these 3, everything I've talked about in the different aspects of this specific disorder included all the different names that you see here. And it would not have been possible without these collaborations between different countries and within a country. And it wouldn't be possible without funding from the National Institutes of Arthritis and Musculoskeletal and Skin Diseases at NIH in the USA. And thank you for listening. Thank you.